Carmen, please.
Welcome back. I'd like to introduce Alexandra Chatzikalymniou, postdoctoral researcher at the Krembil Institute, and she's going to give her talk on the generation of theta rhythms in the hippocampus.
Great. Thank you very much, Carmen, and thank you for giving me this opportunity to present this research talk today, and of course, thank the CCNS organizers for creating this very nice platform to present our work. So I'm going to talk today about work that was mostly done during my PhD about the cellular origin of the theta rhythms in the hippocampus. Now, these rhythms are ubiquitously found in the brain. They are particularly dominant in the hippocampus, where they are associated with spatial navigation, REM sleep, and many cognitive functions. For that reason, they are a very promising biomarker for learning and memory, with applications in many neurodegenerative disorders such as the Alzheimer's and epilepsy. So the idea is that if we are able to decode the information that those rhythms encode during various behaviors, we will be able to know what goes wrong in those pathologies where the features of those rhythms are changed. Now, the C1 area of the hippocampus has this plethora of different inhibitory cell classes, but we still don't have a very good understanding in terms of how these cells contribute to the generation of the rhythms. Now, in my work, I've been using computational modeling to understand the origin of those rhythms, and the experimental equivalent of my models has been this intact in vitro hippocampus preparation that autonomously generates theta and gamma rhythms without any external inputs from the CA3 or the spinal cortex, and without any pharmacological manipulation. This preparation has a number of benefits. The rhythms are not state dependent, they are very regular, and that gives us an opportunity to study the cell types that contribute to them, but also to understand this core CA1 mechanism. And then we can build on it and understand in vivo type behaviors. Now, I've been using two different models. The first one is the biophysically simple minimal model of the CA1, and the second one is the biophysically detailed full CA1 model. The minimal model is comprised of Zikovich neurons, pyramidal cells, and PV-positive cells that are constrained based on data from the intact in vitro hippocampus preparation. The biophysical detailed model is a state-of-the-art model that contains 8 different inhibitory cell types and pyramidal cells, and they are all connected based on experimentally derived connectivity. Now, the local field potential theta rhythm in the detailed model, it is simulated as the summation of the transmembrane currents that pass through the cellular membranes at the vicinity of a recording electrode tip, and they are generated by the pyramidal cells. Now, this takes me now to the first part of my results. In this first part, I'm going to talk about how linking the minimal and a detailed model can help us understand how the theta rhythms emerge and how their frequencies are controlled. This minimal model, we used it in previous studies, which are shown here, if someone is sending me messages about my presentation, because I can hear it on Slack, please speak out if there's something wrong with it. So if not, then probably I'm getting some Slack messages from someone else. Using this minimal model, we found that we generated a hypothesis that says that the resulting theta frequency of population bursts is controlled by the net amount of input that the pyramidal cells receive. And now this is a hypothesis that we want to test to the detailed model. So to do this translation between the models, we first compared them to identify commonalities and differences in their structure. We isolated a piece of the detailed model, the segment model is how we called it, which has comparable cell numbers to the minimal, and that allows us to make comparable, similar comparisons, meaningful comparisons. We found that the two models vary, especially with respect to the excitatory conductances that are driving the pyramidal cells. And now that, of course, could be intuitive in the sense that the segment model has all these other inhibitory cell classes, so probably you need to increase excitation to sustain a certain EI balance of the inputs to those pyramidal cells. But nevertheless, the pyramidal conductances in this model were extremely large. In contrary to the existing literature at the time. And actually, most scientists would think that those connections were not particularly important the pyramidal connections, as they were found to be very rare. So we were intrigued by that, and we wanted to understand why those conductances are this strong in the biophysical model. And so we searched the dependence of the theta rhythm as a function of the ECC3 to pyramidal conductancies and the pyramidal conductancies. So the ECC3 to pyramidal are those external conductances from the external entorhinal cortex and C3 afferents. We can see here that as we increase those conductances, the theta power increases, with the exception of this regime where the network starts producing lower frequency delta activities, and that's why it looks discontinuous. The theta frequency is almost anti-correlated to the theta power. And here I show the stimulation, which is how much I need to activate the pyramidal, it's how much I need to activate those external afferents to get a network that has the maximum theta power that it can give me for a particular configuration of those conductances. We wanted to understand also what is the network doing as we vary those conductances and how the cellular activity is modulated by them. So we look at two cases here, a case A and a case B. One is characterized by low ECC3 pyramidal conductance and the other one by high. And we find that for the low case, because this conductance is low, more stimulation is required to activate the pyramidal cells to a certain amount. We saw this here very qualitatively. And because the stimulation is high, most of the interneurons are also activated in this case. Now, in the case B, because this conductance is already large, we need less stimulation to activate the pyramidal cells by a certain amount. And as a result, because of afferents project to almost all of the interneurons, the activity of the majority of the interneurons is very low. This showed us that likely what is important for the rhythm is the net input to those pyramidal cells, regardless of whether it is delivered by a pathway of a case A type or a case B type. So we looked at this hypothesis more deeply, and we took a sample of 10 cells per network, and we plotted the mean input current delivered to those cells as a function of the theta frequency of the respective network. And we found a very strong correlation between the two. So the net input current to those pyramidal cells could predict the frequency of the resulting theta rhythm. We also found that the inputs to those cells were very noisy, both the ECC3 to pyramidal, but also the inhibitory inputs to the pyramidal cells. This is because this is only a piece of the network, and most of the inhibitory classes had not yet organized into coherently firing populations. But we would still detect the theta rhythm, and that was the interesting part of it, that the theta rhythm was generated by the pyramidal cells, and it was not imposed by some sequential inhibition from the rest of the inhibitory classes. So this is what this segment model allowed us to see. So we further decomposed this net input current to the pyramidal cells, and we found that the strongest was the correlation for the ECC3 to pyramidal input current with respect to theta frequency. There was a weaker correlation for the pyramidal cell input current and an anti-correlation for the inhibitory cell input current. So that's going to take me now to the second part of my presentation. This previous work is now in press in the hippocampus, so there's a lot more figures into it and a lot more results into it that someone can look into if they're interested. But in this next part, I'm going to talk about how we attempted to optimize the segments model according to available biophysical experimental data. So to do this, we now added inputs from the CA1 and the subigulum to the segment model. And this is because this piece, the slice of the hippocampus in a more realistic scenario would also be driven by CA1 afferents and also by newly discovered inputs back projecting to the CA1 from the subigulum. And we get this power frequency plot, which has this really non-specific, I would say, activity. And from there, we focus on the PV positive cells. We don't have experimental data for many cell types, only for the PV and the somatostatin positive cells. But we know that for the PV positive cells, they should fire at the theta frequency, at a frequency that's very similar to the ongoing frequency of the network. So to understand what controls the firing frequency of the PV positive cells, we did a network clamp. So we follow this network clamp approach, in which we vary the postsynaptic weights from the 8 inhibitory presynaptic populations onto a given PV-positive basket cell. And we do this by implementing a grid search. So this is these are just nested for loops that gave us 400,000 configurations. And according to this for loop, the first configuration, for example, would correspond to the first value of the exoxonic cell, postsynaptic weight, the first value of the bistratified cell, postsynaptic weight, so on and so forth, till the first value of the pyramidal cell postsynaptic weight. The second configuration would be the same, only now we move one step forward for the pyramidal cell postsynaptic weight. It's pretty easy to understand. In this iterative way, we vary over all the postsynaptic conductances. And we come up with this plot. Now, this is inherently an 8-dimensional problem, and we have to visualize it in 2 dimensions. So here I'm showing the mean interspike interval of the given PV basket cell as a function of the serial number of the postsynaptic weight configurations. And we can understand this plot if we break it down. So we can see repetitive motifs within repetitive motifs. So the first 5 repetitive motifs corresponds to changes to the exoxonic postsynaptic weight values. So the exoxonics don't affect the PV basket cell firing, so those 5 motifs are identical. If we zoom in to one of these motifs, we can see another set of 5 repetitive motifs. So this now corresponds to incremental increases to the bistratified cell postsynaptic weight. And we can see that the ISI increases a little bit, and this is because the bistratified cells can inhibit a little bit the PV positive basket cells. Now, if we zoom into one bistratified cell postsynaptic weight value, then we can see another set of 5 repetitive motifs. And here's where we see something interesting. For a certain value of the CCK basket cell postsynaptic weight, we see an abrupt increase to the ISI of the PV positive basket cell. And this tells us that if the network sits close to this critical point, a small change to the activity of the CCK basket cell can increase or decrease the firing rate of the PV basket cell even by 20 hertz. And that's something interesting to keep in mind. It's going to be useful later. So according to this grid search, we fitted the postsynaptic weight values to the PV basket cells, and in fact all the other PV positive cells as well. And we now have PV positive cells that fire with a normalized theta frequency very close to 1, which means that the frequency, the PV positive cell firing frequency is very similar to the frequency of the network theta. Okay, so now that's going to take me to the third part of my work. And here I'm going to move from the segment model to the full-scale C1 model. So this is a model that was originally published by Bezer and colleagues in 2016 in eLife. It has this plethora of cell types, and now the different inhibitory classes fire more coherently by virtue of the larger numbers. And the question here is, how is this rhythm generated, but more specifically, what controls this sharp termination of the pyramidal cell theta bursts? So that's something that was not explained in the original publication. The authors did some basic perturbations, but we still don't know exactly what controls this sharp termination of the pyramidal cell theta bursts. So first, let's make some macroscopic observations. We have 2 types of interneurons. The ones that fire at every theta cycle, those are the OLM and the bistratified cells. These are primarily driven by the pyramidal cells. And then we have the rest of the interneurons that are driven by the noisy EC and CA3 afferents. And so they fire more throughout the 4-second period. Let's focus on let's focus to the last 500 milliseconds of this raster plot. Okay, I'm going to exclude a number of cell types that were shown by Bezer and colleagues that do not contribute to theta. And I'm going to focus on the rest. And we want to come up with some hypothesis of how this pyramidal cell bursting is terminated. So here's the first hypothesis that I'm going to present. The pyramidal cells fire, and by virtue of the self-connectivity, more and more cells participate to the theta bursting. Now, the time that it takes for the pyramidal cells to reach a critical amount of activation is defines the theta frequency, and it's defined by the intrinsic properties of the cells and the net input that they are delivered, as I showed in the analysis of the segment model. Now, when they reach a critical point of activation, they turn on the OLM and the bistratified cells. Now, the bistratified cells, they form very strong connections with the CCK basket cells. And so at the peak of the cycle, they inhibit them. So that's the next step. And likely also the OLM cells contribute to this. Now, here's the next step of this hypothesis. The CCK basket cells form very strong connections with the PV basket cells. So the inactivation of the CCK basket cells activates the PV basket cells. And then the PV basket cells terminate the pyramidal cell theta burst because they can now inhibit the pyramidal cells. Okay, let's test this hypothesis to see if this is correct. First, we said that the pyramidal cells activate the OLM and the bistratified cells. So indeed, if we remove the pyramidal to bistratified connections, which are the more important ones, we lose the theta rhythm. So that step in the hypothesis so far seems to be correct. The second step says that the bistratified cells inhibit the CCK basket cells. Indeed, if we remove the OLM and bistratified connections to the CCK basket cells, we don't see that theta-phased quiescence of the CCK basket cells. So that step is correct. However, now that the CCK cells are not inhibited, the PV basket cells remain activated, strongly activated at the peak of the theta cycle. So the CCK inactivation cannot be responsible for the PV basket cell activation, as I hypothesized earlier. So that step of the hypothesis is wrong. But nevertheless, we're going to look at the next steps as well. So the next step says that the CCK basket cells disinhibit the PV basket cells. Indeed, if we remove the CCK to PV basket cell connections completely, we overactivate the PV basket cells. And that's, again, something that came earlier to the grid search, from the grid search of our segment model. So the CCK cells act as PV basket cells controllers. They control their activity very strongly. But nevertheless, and of course, they have the last step, the PV basket cells inhibit the pyramidal cells. That's also true. If we remove those connections, we lose a theta rhythm. The pyramidal cells fire at gamma by virtue of a pink type mechanism. Now, however, the hypothesis that I formulated was not 100% correct. So now I'm going to have to come up with an alternative hypothesis. So this time I'm going to say that the pyramidal cells activate the bistratify and the PV basket cells. The bistratify and the PV basket cells maintain their activity a bit after, for a bit longer, and they terminate, in turn, the pyramidal cell bursting. So it's simply a PV positive pyramidal cell interaction that mediates the termination of theta burst. And I'm going to test this hypothesis now. So in this first step, I said that the pyramidal cells excite the PV basket cells at the peak of the theta cycle. I already showed earlier that they activate the bistratified cells. So indeed, if I remove the pyramidal to PV basket connections, there's no more theta rhythm. The pyramidal cells fire at a constant gamma rhythm, so there's nothing to terminate their activity abruptly. So they have to start organizing into a EE network where they progressively ramp up their activity by virtue of their self connections and the pyramidal conductances. They can't do that anymore, so they just constantly fire at gamma. And then I said that the bistratified cells inhibit the pyramidal cells at the peak of the theta cycle. So indeed, if I also remove the bistratified to pyramidal connections, I see a very similar network to the one that I saw when I removed the PV positive basket cell to pyramidal cell connection. So again, I see a gamma rhythm, a constant gamma rhythm. So then, we said that the PV basket cells inhibit the pyramidal cells at the peak of the cycle, which is the last step of this hypothesis. So of course, if I remove those connections, I lose the rhythm as well. So this hypothesis seems to be much more viable. So that's going to take me now to the end of my presentation. In the first step, I showed that the theta rhythms are initiated by the pyramidal cells using a segment model, and that their frequency is controlled by the net input the pyramidal cells receive. In the second part, I talked about the method of optimizing a PV positive basket cell which showed that the PV-positive basket cells are very strongly controlled by the CCK basket cells. In the third part, I tested two hypotheses that would describe the sharp termination of the pyramidal cell bursting, and I found that the PV-positive to pyramidal cell interactions are the ones that mediate this phenomenon, even in this network that has this multitude of cell types. And I want to bring your attention to this paper that was published recently in Neuron, Alternating Sources of Perisomatic Inhibition During Behavior by Barna Dudok. He showed there that the CCK and the PV basket cells control each other very strongly, and that they're anticorrelated. And he showed that in numerous behaviors in vivo. And that was super interesting because that spoke exactly to the results that I just showed you now in this model. So we have experimental evidence that the CCK control to the PV basket cells is experimentally realistic. So thank you very much for your attention. I would like to thank my supervisor in the Skinner Lab, my supervisory committee members, and of course, NSERC, the Alexandra Graham Bell Scholarship that supported a big part of my PhD work.
Thank you, Alexandra. Great talk. Carmen, do you want to take care of the questions?
I'm toggling. Oh, there. Okay. Sure. There's a question from Scott in the questions that the division between the two interneuron firing patterns seems to match, whether they're somatic or dendritic targeting. How might that be affecting theta development? And I guess we're thinking in terms of the dipole. Any comments, Alexandra?
Right. So is the question about how the inhibition at the different layers would affect the polarity of the LFP, so this current source density profile? Maybe Scott, I didn't get the question right.
Yeah, let's see. And there's another question from John Rubin. From part 3, do you conclude that most of the inhibitory neuron types are not important for the theta rhythm? And I was a little confused on that too, because in the first part you say the pyramidal neurons initiate the rhythm and control the frequency, but then in part 3 you're showing that if you block these inhibitory neuron types, you abolish the theta rhythm. So like I said, John's question is, you conclude most of the inhibitory neuron types are not important for the theta rhythm.
So we in the first part, I showed that the pyramidal cells on their own can generate a very baseline theta rhythm. It's not as regular and it's very noisy, but it is there. So the idea that I was trying to convey is that the theta rhythm sparks off by the pyramidal cells, but then the inhibitory cells contribute to the robustness of the rhythm. And that robustness of the rhythm is mediated exactly by this precise temporal control of the termination of the pyramidal cell theta bursts at the peak of every theta cycle. And the cells that so far I have seen to be important for this are the PV positive cells, but also the CCK cells, in the sense that they control the PV positive basket cells and they make sure that the network is situated at a dynamical state where the PV positive basket cells are not overly active. So yeah, so this is the prediction.
So is there a contribution of intrinsic oscillation in the pyramidal cells, or is it all network?
So I would say that the frequency, I can describe it this way. The time it takes from here to here, so the time, so let's take a particular example, a particular cycle. The pyramidal cells are silenced here. So then they are driven by those noisy afferents, and they start building up their activity. So how fast and how quickly they're going to build up their activity to reach the critical point where they start activating other inhibitory classes, that time is going to be defined by the intrinsic properties of the pyramidal cells. But once they do that, once they activate the PV positive cells, then the PV positive cells take over, they terminate them, and then another cycle starts again. But this duration right here is defined by it's very strongly defined by the intrinsic properties of the pyramidal cells. And I have evidence, for example, that IH plays a huge role in this.
And is that nested gamma bands that I'm seeing in the?
Yes, exactly. So there is also a theta gamma phase amplitude coupling phenomena going on here. And we can explain why there is coupling of the gamma amplitude and the theta phase, in the sense that by virtue of pyramidal interactions with other gamma firing cells, this could be the CCK, the PV basket, even the Schaefer collateral associated cells or the exoxonic cells. By virtue of those interactions, you have a pink rhythm. So you have a gamma rhythm on the pyramidal cells. Now, as the activity of the pyramidal cells increases, then the gamma power increases. And so because the participation of the pyramidal cells is maximum at the peak of the theta cycle, that's where the gamma amplitude is maximum as well. So that's why you have theta gamma phase amplitude coupling.
Okay, thank you. And I think we probably need to move to the next talk.
Yes, thank you so much, Alexandra and Carmen. Stay with us for the keynote talk by Professor Chatzikalymniou.